Climate change is one of the biggest challenges in the field of geosciences. The Genus project is investigating the effects of climate change on the Benguela upwelling system off the coast of Namibia.
Die Folgen des Klimawandels auf der Erde betreffen nicht nur das Abschmelzen.
The effects of climate change on the Earth not only involve ice melting and sea levels rising, but also changing wind systems around the globe. And one of these wind systems is the trade wind. The trade wind is a wind motor for currents around the continents, for example off southwest Africa, and stimulates the upwelling, as it's known, of cold, nutrient-rich water. In this coastal area off the states of Angola, Namibia and South Africa, the trade wind constantly blows along the coast and drives the surface water towards the ocean. Cold water comes to the surface and brings with it unbelievable amounts of nutrients, which are the basis of an extremely productive ecosystem. The Genus project is funded by the German Federal Ministry of Education and Research and combines the expertise of 7 of Germany's marine research institutes, scientists specializing in physics, chemistry and biology. It also involves cooperation with Namibian and South African scientists. At the University of Hamburg, we are investigating the nutrient cycle in this upwelling water, looking at the gas exchange between the ocean and the atmosphere. And one group is dealing with zooplankton, an important link in the nutrient chain in this area. The University of Hamburg is also coordinating the whole project, because we in Hamburg are at the center of this group and are aiming to treat the various aspects of this upwelling ecosystem as a single unit, and as geochemists also to work on it scientifically ourselves.
The coordination of an international research project also includes the planning and execution of marine expeditions. First, a request has to be put in for ship time. The neighboring states must give their permission to enter their territorial waters, and all the equipment has to be packed and shipped in good time. In the process, a research ship is transformed into a floating laboratory, tailored to the needs of the individual projects.
Im Projekt Genus führen wir mehrere Schiffsexpeditionen vor der Küste Namibiens.
In the Genus project, we are carrying out several marine expeditions off the coast of Namibia. And of course, we have to plan and organize extensive logistics for this, as we have several sub-projects dealing with physical, oceanographic and biological subjects. This means we will be sending up to 6 containers to Namibia, all completely packed with equipment, i.e. machines, instruments and consumables. A total of several million euros worth of equipment for our expeditions will be sent in the containers to the respective ships. Of course, it's important to remember everything in the planning, as whatever is not on the ship will not be available. And sometimes small things like a cable or a screw could then be crucial to the success or failure of a project. On top of that, there are also numerous hazardous items that are shipped requiring specific logistics, which then all has to be implemented by the Genus scientists themselves, and then all the threads come together at the coordination facility in Hamburg.
Seen from a global perspective, the Benguela upwelling system is one of the biggest and most important upwelling systems. A special feature of the Benguela system is the wide continental shelf. The area is bordered by warm water masses to the north and to the south.
You can see on this graph effectively a conceptual model of roughly how this system works, although it is quite rough. Many processes which play a role here are not shown in the model, but the basics are. Here we have the coast of Namibia, and we have a southeasterly trade wind which has a component parallel to the coast. This component drives an Ekman transport, which is represented here by the red arrow. The water that is on the coast here is pushed away from the coast out into the open ocean. This means we get a lowering of the water level here, and this is compensated for by water rising from the depths. And this water has to come from somewhere, and that is the open ocean. This water is very nutrient-rich and effectively pumps nutrients from the open ocean onto the shelf.
Inorganic nutrients like nitrate, phosphate and silicate are thus transported from the depths to the water's surface. The distribution of these nutrients on the Namibian shelf largely forms the basis of where along the coast high levels of algae are produced in the sunlit zones of the water. The inorganic nutrient cycle is one of the key subjects that Genus Project's biogeochemical study team is focusing on. The oxygen concentration in the water column is also being investigated. Areas where there is little or no oxygen are found in a number of places on the Namibian shelf. The presence of these suboxic and anoxic areas has a considerable influence on the organisms living in the Benguela system. The constant gas exchange which takes place between the atmosphere and the ocean is also of interest. Research is being done into which zones of the shelf of Namibia act as sources and which act as sinks for atmospheric CO2. Biologically high productive zones like the Benguela upwelling system, which are characterized by enormous growth of phytoplankton, are able to store large volumes of greenhouse gases. This so-called biological pump works against the physical pump. The Benguela upwelling system is therefore ideally suited to research into the role of upwelling systems in relation to regional and global climate change. The primary production stimulated by inorganic nutrients forms the basis for the nutrient chain in the ocean. The phytoplankton is consumed by zooplankton, by copepods and other small crustaceans like shrimps or krill. This means fish also find enough nutrition. Herring-type fish like sardines and anchovies, large predatory fish, and even marine mammals like dolphins and whales. The plankton is therefore of key importance for the understanding of nutrient networks in the Benguela upwelling system. Samples are being taken at a large number of sites with various types of net using varying depths and biogeochemical framework parameters. The biologically oriented sub-projects investigate the phytoplankton and zooplankton groups in particular, which are especially significant for understanding the Benguela ecosystem. Small crustaceans like the euphausiids, the krill, are an important indicator for the ecosystem of the African coast. The 8 known types of krill in the Benguela system, like many other types of zooplankton, are located in various sections of the project area, depending on the physical and chemical conditions. And even the day-night cycle has an impact on the water depths at which the individual species are caught in the nets. For that reason, it is often necessary to estimate in advance at what depth the nets should be deployed. Only when you have sufficient sample material available can you continue with the experiments on board in the ship's laboratory. Fish larvae and small fish, so-called ichthyoplankton, are also usually found where krill and other types of zooplankton are located. However, a dramatic change has occurred in recent years and decades. Both the size and the composition of the fish population appear to have changed significantly.
Ja, es hat sich einiges in den letzten Jahren verändert.
Yes, there have been some changes in recent years. Usually, it is the sardines and anchovies which are, let's say, the primary predators in these upwelling systems, which eat all the zooplankton, the small plankton and the phytoplankton, and which can use this basic food source to build up large stocks. The problem these days is that there are no sardines. The current stock is very small, yielding maybe 10,000 tons a year, which is really nothing compared to the 300,000, 400,000, 500,000 tons that there used to be in the good old days. And the question is, why aren't they there?
The changes also have significant socio-economic effects on the whole region in the south of Africa. The whole Namibian fishing industry is feeling the profound change in this highly productive system. Projekt Genus is currently investigating the precise causes. Fish stocks have fallen, but the resulting gap in the system could perhaps already be closing. Other organisms appear to benefit from it. They occur in swarms and have only a few natural enemies. jellyfish.
Diese Quallen, das sind auch räuberische Organismen.
These jellyfish are predators which feed on the same organisms as fish. Both prey on zooplankton. And if the fish stocks fall, there is more food available for the jellyfish, and they are able to multiply more. And so yet another impact of climate change comes under discussion. This is not yet completely proven, however, and we hope to get an insight into this with our project.
There are various factors which must be recorded. A change in each individual variable can have an effect on the overall structure. In the Genus project, the data captured are to be entered into a mathematical model which displays the complex relationships. What happens, for example, if the trade wind, the motor driving the upwelling, gets weaker? Hypotheses like this are to be tested in the model.
We can test this with the model. We can already do this. We've got that far. But of course, we don't yet know whether we have actually adequately represented all the very important processes that play a role in this special marine area in the model. This is something we still have to test carefully. It might well be that there are still a few surprises to come and things to learn, things we may have missed. We still have work to do, and that's why we're taking so many measurements.
A central objective of the Genus project is to network with international partners and research institutions, in particular involving regional scientists from states bordering the Benguela region.
Dieses ganze Projekt ist nicht isoliert, sondern wird betrieben im Rahmen.
This whole project is not isolated, but is being run first and foremost alongside internationally collaborative projects and internationally coordinated projects. For example, IMBA, Integrated Marine Biochemistry and Ecosystem Research. In this international project network, the question is, just like in the Genus project, how the changing climatic conditions are affecting material cycles, CO2 cycles between the ocean and the atmosphere, and how this changing circulation pattern will affect the structure and viability of marine ecosystems. At the same time, in the internationally coordinated LOIX project, Land-Ocean Interactions in the Coastal Zone, there are a whole range of interesting projects which focus on shelf seas. Our Genus project is on the dividing line between these two international projects, IMBA and LOIX, and attempts to answer these interesting questions using the example of the coastal upwelling system of Southwest Africa.
On the research vessel Discovery, a microstructure probe is being prepared for use. The turbidity sensor is adjusted to the clean oceanic waters off the coast of Southwest Africa. As part of the Genus project, Volker Moorholz and Thoralf Heene are analyzing the physical processes in the seas off the coast of Namibia. The focus of the research activities is to investigate the influence of climate change on the water masses in the Benguela upwelling system.
The Benguela upwelling system is one of the world's 4 great upwelling systems. These systems are always on the western edges of continents or eastern edges of the oceans, usually in subtropical regions. We have one here off Southwest Africa, the Benguela upwelling system, then one off Northwest Africa, then off Chile, the Humboldt upwelling system, and finally the California upwelling system off the west coast of the USA.
One unique feature of the Benguela upwelling system is the wide continental shelf. The area is also bordered to the north and south by warm water masses, which is not the case for the other 3 large upwelling systems.
If we look at this picture, here we have this large vortex in the South Atlantic. We call this the subtropical vortex, which constantly churns the water around in the South Atlantic. And here in the south there is water from the Cape Basin, which is here, while from the north we get tropical water in the same area from the equatorial current system. This is the area we are mainly working in, and this area is essentially a transitional zone between two large central water masses, as we say. At one point we have the South Atlantic Central Water, which has specific characteristics. It is very low in oxygen, but therefore very nutrient-rich. And from the south we get a second water mass, the Eastern South Atlantic Central Water, which is rather low in nutrients, but for that reason oxygen-rich. And from the interaction between these two water masses we get the characteristics that we have here off northern Benguela. We also see strong seasonality here, that is to say, this oxygen-rich water mass comes into the area with the upwelling in the austral winter. Meanwhile, in the austral summer, a water mass low in oxygen comes into the area from the Angola vortex and then also has very powerful effects on the ecosystem.
The different water masses are relatively easy to identify based on temperature and salt content, but that alone is not enough. Current speeds, oxygen and nutrient content also play an important role and provide information about the state of the system. Numerous key parameters important for the Genus project can be measured directly on board with so-called online measuring systems like a ferry box, for example. However, this only enables the composition of the water's surface to be analyzed. For that reason, additional measuring equipment is used at the various stations in the study area. The main device used by the physical oceanographers is the CTD rosette. This can measure the temperature, the salt content, and some additional parameters of the water at a depth of up to 3,000 meters.
CTD bedeutet, zusammengesetzt aus 3 englischen Wörtern.
CTD is a combination of 3 words, C for conductivity, T for temperature, and D for depth. It is a device which measures all 3 of these and other parameters too, but this is a classic CTD which measures these 3 parameters, and then figures derived from this, like salt content and density, can be determined from the conductivity and temperature. We also work with other study teams like the chemists and biologists who need water for analysis. And for that purpose we have these bottles. They are simply tubes, and when the CTD is lowered they fill with water, or water flows through them, and we can close the bottles from our control panel in the laboratory. I do it like this. Then the section of the water column is captured. And then you have, in this case, 10 liters of water for analysis in the laboratory. We have also combined this with our current profilers. These are acoustic current profilers which are able to measure water currents at, off the top of my head, about 100 meters, once from the top and once from the bottom of each device. So we can determine the current for approximately 200 meters from the CTD. And as the CTD moves from the water's surface down to the seabed, we always have overlapping 200-meter profiles, which we put together afterwards, and we get a current profile for the whole water column.
Another measuring device is a microstructure probe. Like the CTD, it measures the temperature and salt content of the water, but can also detect current shear and small vortices in the water. The device, weighing around 50 kilograms, is deployed to free fall slowly and is balanced so that it sinks down independently and measures vertical profiles with a resolution of 2 to 3 millimeters. Sensors are installed at various depths using moorings that stay in the water for several years. These sensors measure all the important physical parameters of the water column several times a day. The resulting measurements therefore give a high-resolution temporal and spatial picture of the water mass in the study area. The physical oceanographers in the Genus project are investigating hydrographical environmental factors and documenting their change over a long period of time.
We have been working here for around 10 years. We've already investigated the basic processes in the ecosystem in previous projects. However, some specific processes which have an important influence remain unresolved, and we now want to investigate these. In recent years, it's been shown, for example, that in the Angola vortex, there is a temporal trend in oxygen content. That is to say, the oxygen has slowly decreased over the last 50 years. And the question is how this fall in oxygen content in the water, which is being transported from there into this system here, will affect the entire ecosystem.
My name is Maike Rüschkamp. I studied geosciences at the University of Hamburg, and this is now my second trip as part of the Genus project here in the Benguela upwelling system. Here on board, I'm researching material flows within the sediment and between the sediment and the water column above, looking at nutrients, dissolved gases, and dissolved metals and trace metals. For my research here on board, I need sediment cores. These are obtained using a multicorer equipped with 8 plexiglass tubes. The multicorer works purely mechanically. We can use it to take samples at water depth of several thousand meters or to sample the seabed at such depth. The multicorer is attached to a cable and lowered to the seabed. There, the 8 plexiglass tubes bore into the sediment. When the device is raised slightly, the plexiglass tubes automatically close at the top and bottom, so that the sediment cores are brought to the surface as undisturbed as possible and in one piece, and are then ready for further investigation. This multicorer concept works well in soft and silty sediment. In hard, stony sediment, the tubes cannot penetrate the sediment and therefore return to the surface empty. When analyzing the sediment cores, I begin by measuring the oxygen. I measure the oxygen content and the oxygen penetration depth. So I look at how many centimeters down the sediment we find little or no oxygen. sauerstoffarm bzw. sauerstofffrei zu werden. Als zweites kommt dann die Analyse. Secondly, I analyze the dissolved gases in the sediment core. To do this, I use a membrane inlet mass spectrometer. The inlet of this mass spectrometer is this needle. In front of it is the membrane, so a small opening. The advantage of this device is that I can place this needle directly on the surface of the sediment core, that is, at the transition between water and sediment. And then I can measure the gases directly within the sediment core. So I will start now. I put the membrane on the surface of the sediment. So, jetzt sitzt die Membran an der Sedimentoberfläche. Now the membrane is on the surface of the sediment. Now I can switch on the computer that controls this needle so that it bores into the sediment in 3 mm stages. At the same time, I then measure the gases using the mass spectrometer. Als weiteren Schritt meiner Analyse untersuche ich die In another stage of my analysis, I investigate the nutrient concentration in the supernatant water and in the pore water, which is the water between the individual sediment particles. To do this, I put this little filter into the sediment core. As you can see, the sediment core has already been prepared accordingly. There are holes at 1 centimeter intervals, which were covered when the sample was taken. Now I can put my filter in here and get a depth resolution of the nutrient concentration. Now I take supernatant and pore water from each depth down to 10 centimeters into the sediment core and analyze the nutrient concentration later in the laboratory. Ammonium, phosphate and so on. I have already prepared everything for this sediment core. I unscrew the little cap and can attach my syringe. I create a vacuum in the syringe, and the water moves through the filter into my syringe. Later I put the water into suitable vessels and prepare it for transport to the laboratory at home. After 14 days of research here in the Benguela upwelling system of Namibia, we have sampled different water depths and obtained different sediment cores. Here I have arranged a selection of different sediment cores. We have a sediment core from a depth of 230 meters. This comes from the shelf. We have a sediment core from a depth of 830 meters from the upper slope, and a sediment core from a depth of at least 2,000 meters from the mid-slope. You can clearly see the different sediment types from the different depth. In the sediment core from the shelf, you can see individual pieces of crushed shells, as well as this greenish-brown sediment. This is probably partly made up of dead, decomposed phytoplankton. In the middle sediment core from the upper slope, you can see that it is considerably different. It is rather sandy sediment containing very little organic matter. Then we have these dark spots in this sediment core. This is probably from the low oxygen and oxygen-free zones. Then we have the sediment core from a depth of at least 2,000 meters, this grayish sediment. My colleague has already looked at this under the microscope. It is almost exclusively made up of carbonate shells from dead microorganisms.
Mein Name ist Matthias Birkicht. Ich bin beschäftigt am Zentrum.
My name is Matthias Birkicht. I work at the Center for Tropical Marine Ecology in Bremen. I'm currently on board the research ship Meteor, along with my research group from the biogeochemistry sub-project with Anita Flohr and Tim Rixen. I'm working on nutrient analysis, which is a basic parameter for the evaluation of nutrient flows in the ocean.
Im Ozean darstellt. Wasser. Als Elixier.
Water, the elixir of life, contains nutrients which, here in the Benguela upwelling system, are transported upwards from the seabed by cold ocean currents.
Es handelt sich bei den Nährstoffen
The nutrients include phosphorus as phosphate, nitrite, nitrate, and ammonium.
als Stickstoffkomponenten,
as nitrogen components, and the silicon important for diatoms. The algae on the ocean's surface consume the nutrients transported upwards and, in doing so, feed the organisms further up in the food chain.
weiter oben stehenden Organismen.
In order to research the distribution of these nutrients in the water column on the seabed and in the sediment,
Zu erforschen, bedienen wir uns auf der Meteor
we use a variety of devices on the Meteor.
Geräte. Ein Gerät
One device which samples the different depths in the water column is the CTD rosette.
die CTD-Rosette. Das ist die CTD-Rosette.
This is the CTD rosette. In the center, it contains a physical measuring unit which records temperature, conductivity, and depth.
Sobald die CTD-Rosette
As soon as the CTD rosette is lowered into the water, we are able to close each of the 14 individual water samplers so that they retain the water that is scooped up, bringing it to the surface.
was geschöpft wurde, behalten. Nachdem
After the rosette has completed the journey to just above the seabed and all the samplers have been closed, the CTD rosette is lifted back onto the deck.
gehoben. So, nun haben wir ja schon fast
Now we have sampled almost the entire water column with the CTD.
Problem stellt der
The 1 meter above the seabed presents a problem because the CTD rosette cannot be placed on the seabed. For this, we use a very special device, the bottom water sampler.
Bodenwasserschöpfer. Der Bodenwasserschöpfer hat 5 parallel.
The bottom water sampler has 5 Niskin bottles, arranged in parallel at different levels, which are first opened and then remain cocked and ready.
Gespannt bleiben. Sobald der Bodenwasserschöpfer
As soon as the bottom water sampler rests gently on the seabed,
schließen sich
the lids of the Niskin bottles close and seal the water from the seabed in the bottles.
in den Flaschen fest. Nun fehlt uns ja nur noch der Meeresboden.
Now, all that's missing is the seabed.
Besteht aus Meeressediment. Locker aneinander gebunden.
The seabed consists of marine sediment and particles loosely bound to each other, which contain pore water.
Ganz dicht über dem Meeresgrund
Just above the seabed, we have the supernatant water. In order to collect the supernatant water and the pore water, we need a special instrument, the multicorer.
Multicorer. Der Multicorer arbeitet mit
The multicorer works with core cutters, which are fixed into the mount.
Wenn der Multicorer den
When the multicorer reaches the seabed, the weight rams the core cutters into the seabed and gathers a sediment core with supernatant water.
Überstandswasser. Die Rohre
The
werden
tubes are firmly closed at the top and bottom, and the multicorer is lifted onto the deck.
gehoben. Ich befinde mich jetzt mit all meinen Proben im Labor.
Now I'm in the laboratory with all my samples, which I transfer into the sample injector on the auto-analyzer. For each individual nutrient to be analyzed, we need reagents. These reagents are mixed with the sample in the system.
Vermischt. Da eine Reaktion Zeit
As a reaction takes time, we have long spirals where the sample can react with the reagent.
reagieren kann. Während der Reaktion
During the reaction, a color develops which is specific to each of these individual nutrients. Nitrate, nitrite, ammonium, phosphate, and silicate.
eine der Farbreaktionen
We will simulate one of the color reactions for the nutrients in this test tube.
das Reagenz
We add the reagent to the sample,
und die Reaktion setzt ein.
and the reaction takes place.
Anhand der
The analyzer can accurately determine the concentration of the nutrient based on the intensity of the color.
Nährstoffes bestimmen.
Mein Name ist Anita Flor. Ich bin Doktorin.
My name is Anita Flor. I'm a postgraduate student at the Center for Tropical Marine Ecology in Bremen. And as part of Genus, I'm working towards my doctorate, looking at the nutrient cycle, but predominantly the carbon cycle here in the Benguela upwelling system. My main tasks during an exhibition like this include taking numerous water samples. I take these water samples from the bottom water sampler and the CTD rosette. The bottom water sampler is used to sample the 1.5 meters of the water column above the sediment. And in contrast, the CTD rosette is designed to scan and sample the entire water column.
Zu scannen und zu beproben. Ja, die CTD-Rosette ist für die Untersuchung.
The CTD rosette is a very important device for the research I want to carry out. CTD stands for conductivity, temperature, depth.
Leitfähigkeit, Temperatur, Tiefe.
And the CTD will now be lowered through the entire depth of the water down to just above the seabed, and effectively records an online profile of the oxygen content, the temperature and the salinity. This site here is around 360 meters deep, but we could work at a depth of up to 5,000 meters with a CTD rosette. So on this monitor we can effectively follow online as the depth profile of the individual parameters is displayed. Each site shows a completely different profile, and depending on these profiles I then decide what depth I want to sample. And now you can briefly tell me what depth you are sampling for me. Which ones would you like? I would like the 30 and the chlorophyll maximum. Now the CTD rosette is back on deck, the water samplers have closed, and now I have to double check that this is the correct sampler. Number 12. I have also prepared various sample bottles for the different parameters I want to measure. These ones here, for example, are exotainer tubes. I now have to fill these without any air bubbles, and then later the dissolved inorganic carbon will be determined. A good rinse. And check that it is free of air bubbles. This bottle here is filled with a sample to determine the alkalinity. This also has to be rinsed first. And along with the results of the dissolved inorganic carbon, we can then determine the carbonate system from these two parameters. Then we can state how much CO2 is dissolved in the water and how acidic the water is. I fill this bottle with water for the nutrient analysis and for determining the DOC. DOC stands for dissolved organic carbon. The water samples are now processed in different ways. The samples are sometimes only measured after 2 or 3 days, or only after the expedition back in Bremen. In order to ensure that the samples are kept exactly as they were when I filled them, they have to be fixed. We can do this by poisoning or freezing the sample. This stops all the biological processes that could have an influence on the parameters I want to measure. The samples for determining the alkalinity and the analysis of dissolved inorganic carbon, for example, are poisoned and are measured here on board. The samples for the analysis of dissolved organic carbon are frozen and then measured back in Bremen. The nutrient samples are measured on board straight away, so fixing is not necessary. The only thing that has to be done first is to filter the water sample. By pushing the water through this filter here, all of the minute organisms that are larger than 0.45 micrometers in size are trapped in the filter, and this cleans the sample, which is then ready for analysis. When the nutrient samples have been analyzed, we can create profiles. What we can see here is an oxygen profile, a nitrogen profile, and a phosphate profile. And what is very characteristic of the Namibian shelf are these distinct oxygen minimum zones. These are shown here in red. This is an area in which the amount of dissolved oxygen in the water column is very low. This is in contrast to sites away from the coast, where we measure very high oxygen values. And what happens in these oxygen minimum zones on the shelf here is that nitrogen is lost from the water column through processes such as denitrification and anamox. This means that we measure moderate nitrogen concentrations above the shelf, but the maximum values are usually away from the coast and at greater depth. For phosphate, the situation is actually the other way round. Phosphate increases greatly in these oxygen minimum zones. And this is actually due to the activity of bacteria that live in oxygen-deficient conditions and add phosphate to the water column. The result is that the ratio of the two nutrients changes significantly, and this variant nutrient ratio is then brought to the surface during the upwelling and transported into the open ocean and to the South Atlantic, with repercussions for the nutrient balance in the South Atlantic.
In the Genus project, we are investigating the material and element cycles in the Namibian upwelling system. This means we take samples from the ocean's surface with nets in order to investigate primary production, and we also go down to the seafloor with sediment devices and take sediment samples. But in order to be able to describe and analyze the material transport, we need the vertical material transport through the water column. To do this, we deploy so-called sediment traps in the ocean, which measure the vertical particle flux in great detail for us over the course of 1 year or so, and provide us with important information about the material cycle in the Namibian upwelling system.
Here
we have an example of a sediment trap which we deploy in the ocean. In this case, it is a relatively small trap as we use it in shallow areas. If you imagine that the system is installed in the water column, at the top here it has buoyancy floats and it also continues below. Assuming we have a water depth of 200 meters, then at the bottom here we have an anchor on the sea floor at 200 meters, and this device then hangs at a depth of around 100 meters and measures the vertical particle transport through the water column. You can imagine that the particles sink from above and trickle down. And then here we have the funnel. The particles fall into this funnel and then into the sediment trap. This funnel takes them in, the particles continue to fall through, and then fall into the first sample bottle which is under this funnel. The sediment trap is programmed to rotate the sample bottles once a month, so that the vertical transport for each month can be collected in these bottles. When the sediment traps have been brought back on deck after a year or more, the bottles are unscrewed and can be used in the laboratory for qualitative and quantitative analyses.
Für qualitative und quantitative Analysen weiter auswerten.
After the trap has been recovered, we take the trap bottles into the laboratory and carry out the first chemical analyses, whereby we investigate the pH value, the oxygen content, and the salinity in order to record general parameters for the sedimentation process. Then we pour the material of each cup into a tray and investigate the composition on a macroscopic basis. This means we look at what zooplankton and phytoplankton are present, and any abnormalities are also noted, of course. Depending on the zooplankton and phytoplankton communities present, conclusions can also be drawn about the sedimentation process during the time the trap was deployed. And of course, we can also immediately see whether there are any unusual organisms present. There are often many species of krill and copepods, which can of course be identified in order to understand how the sedimentation process, and indeed the whole ecosystem, has functioned over the year in the upwelling system. After we have finished our macroscopic description, we filter our samples using polycarbonate filters and dry them for 24 hours at 40 degrees in a drying oven. Then we can very quickly carry out a qualitative and quantitative analysis of the sedimentation processes in the area we are working on. We can instantly see that the community formation of the plankton species change somewhat throughout the year. To start with, we mainly have diatoms and similar phytoplankton. And if we move further into the year, we can see, for example, that we have small crustaceans. And if we look at August, September, October, in some cases there are a few fish scales and a few snails. So we can see that the sedimentation varies greatly throughout the year, both qualitatively and quantitatively. We then carry out further analyses back in our home laboratories in order to be able to better examine biogeochemical aspects of the sedimentation process and to ultimately complete a biogeochemical description of the material cycles. Here we are in Laboratory 6 of the research vessel Meteor off the coast of Namibia in the Benguela upwelling system. My name is Tim Rixon. I am the head of the Carbon and Nutrient Cycles Research Group at the Center for Tropical Marine Ecology in Bremen. And I am also a visiting professor for biogeochemistry at the University of Hamburg with the aim of better combining research and teaching. Here on the ship we are attempting to broadly analyze the carbon cycle. This means we measure the carbon in the atmosphere, in the water column and in the sediment. We have devices that measure the carbon concentration in the atmosphere and in the water, and we work together with other colleagues who operate the CTD. We take water from the water column and determine the carbon content there, and we take samples from the bottom water sampler, which collects water close to the sediment. We also sample the sediment itself and investigate the carbon cycle in the sediments, specifically in the pore water and in the solid phase. So we are measuring atmospheric carbon, water column carbon and sediment carbon, and we are attempting to quantify and track this entire flow from the atmosphere into the sediments. One of the most important transport processes we are familiar with is photosynthesis. We all know photosynthesis is what green plants do. They effectively take CO2 from the air, and then, put simply, it reacts with the water to form organic material, which is the biomass. This can be abbreviated as CH2O, and in the process oxygen is produced, which is why we say that green plants are the lungs of the earth. What happens is that the carbon, the CO2, which comes from the atmosphere or is dissolved in the water, is converted into organic material. This sinks through the water column and is then stored as organic material in the sediments. However, some of the carbon is degraded again, respired by bacteria and comes back here as CO2. Our task is to find out how much CO2 comes from the atmosphere and how much CO2 comes from the deep ocean. The special thing about the coastal area off Namibia is that it is one of the 5 areas of the ocean that are particularly productive. Here we have what are known as eastern boundary current upwelling systems, the huge upwelling areas off California, the Humboldt system off Peru. Here off Mauritania we have the Mauritanian upwelling system. Here in the Arabian Sea we have the upwelling system off Oman, off the Arabian Peninsula, and one of these 5 is the Namibian upwelling system, the Benguela upwelling system. These upwelling systems are characterized by incredible biological productivity, and this is something we've already seen. During photosynthesis, CO2 reacts with water to form biomass. Huge amounts of biomass are produced, and oxygen is also released, but this oxygen escapes into the atmosphere. The organic material, however, sinks into the deep sea, which has no direct exchange with the atmosphere, and this is where all the organic material decomposes. When organic material decomposes and there is no oxygen present, it begins to smell. It's like rotten eggs which smell of H2S. That is the substance that causes this smell. But it's not just H2S that forms. Bear in mind there is no oxygen there, and there is little or no oxygen in this zone, which partly applies right down to the sea floor. This means the whole water column is very low in oxygen. If you have no oxygen, you cannot form any CO2 either because it contains oxygen. Instead, CH4 is formed, which is methane. Methane contains no oxygen. However, methane also rises to the surface where it is released into the atmosphere, and as we know, methane is a much more potent greenhouse gas than CO2. This means it isn't just about the CO2 balance. Depending on the oxygen content in the water column, we must also decide how much CO2 and how much methane is produced and escapes into the atmosphere. This is crucial to our understanding of the greenhouse gas effect and our future climate change, as some studies have already shown that these oxygen minimum zones are expanding globally and we may get more methane than we had before. The main problem is that we are subject to strong seasonal variability, which means when we are here in January, the conditions are very different to in February or March. This means you actually have to be here in the field continuously in order to be able to understand these processes better and to be able to really quantify them. In addition, everyone knows we live in an age of global change, which means year-on-year variability is of great significance. Of course, we cannot do everything on one or even a few expeditions, but the Meteor has been traveling in the area around Africa continuously for the past 6 months. This means our devices have been running continuously here for that time, and each trip has had 1 or 2 members on board in order to get some degree of control over the variability. There are also other colleagues with whom we work, of course, from the Max Planck Institute in Jena, for example. They have built a so-called greenhouse gas tower near Gobabeb, which is situated in the north of the Namib Desert. Here they will measure greenhouse gases for several decades, and our research will help with understanding the marine contribution to the changes that are measured there. To sum up, here we are in an upwelling system off Africa. So here we have Africa, then we have the African shelf, which goes down into the deep sea. Here is the water surface, and the upwelling is characterized by the fact that we have deep masses of water which are laden with CO2 and nutrients which come to the surface here. Where the upwelling water comes to the surface, we have noticed that we have ridiculously high CO2 emissions. But when we move further away from the coast, these nutrients are used by phytoplankton and CO2 is absorbed. The ocean is undersaturated, so it absorbs CO2, and far out into the open ocean, the CO2 concentrations are similar to those in the atmosphere. The difficulty we have now is seeing how much CO2 is released here and how much is absorbed. This is what we're trying to balance. However, due to the fact that we have oxygen minimum zones here where there is no oxygen present and we have methane, methane also comes to the surface, and we have noticed that there is some supersaturated methane here. But where the biology thrives, where the zooplankton eats the phytoplankton, it is similar to where you have cows on land. A lot of methane is produced and the methane goes up. This means we have a zone where CO2 is absorbed and a zone where methane is released. And we have to balance this out, which is not that easy. You cannot do this from the amounts, as methane is a more potent greenhouse gas than CO2, so we have to look at the amounts and the greenhouse effect, which is what we will do as soon as we get home.
Norbert Wasmund and his colleague Anja Hansen are studying phytoplankton in the Genus project. The cyanobacteria are of particular interest, also known as blue-green algae. They're not found in high abundances in this area up to now, but they might become more important with climate change. On board the research vessel Discovery, barrels are being prepared for an experiment. The intention is to investigate the specific conditions for cyanobacteria growth.
Plants need nitrogen to build proteins, for example. Gardeners spread it onto their gardens as fertilizer. And algae in the sea also need this nitrogen. They are often limited in terms of nitrogen, and the cyanobacteria, which are able to use and fix nitrogen from the air, are an important link in making this nitrogen available to the ecosystem.
Nitrogen is present in the air, but many plants can only utilize nitrogen in the form of inorganic nitrogen, as ammonium or nitrate. Cyanobacteria are one of the very few organisms which are able to convert atmospheric nitrogen straight into organic nitrogen compounds. This process is called nitrogen fixation.
We want to investigate whether nitrogen fixation takes place here in this area of Southwest Africa, of Namibia and Angola. Nitrogen fixation does not take place uniformly across the globe, but only in specific regions under specific conditions. In theory, the conditions are right here, but nitrogen fixation has not yet been proven here in practice. If these special nitrogen fixers have not been found previously, that does not mean that they are not present, but they are probably just here in small numbers.
Good growth conditions for the cyanobacteria are being produced in the barrels. By this means, it can be ascertained whether the cyanobacteria are present and what conditions they grow in.
The cyanobacteria make the nitrogen they need themselves, but they need phosphorus. That is to say, if I give them more phosphorus, they are able to grow better. They also need other conditions, for example, iron and various vitamins. If I make these available, they should essentially grow, unless, of course, they are not there in the first place. We want to establish this in the barrels, whether they grow under favorable conditions, even at higher temperatures, for example here. If they do not do this, then they are probably not present. In the open ocean, most of the cyanobacteria are of the genus Trichodesmium, the nitrogen fixers. They are filamentous cyanobacteria which actually occur in bundles and are therefore very noticeable. There are, however, possibly also nitrogen fixers which are very inconspicuous, small coccoid cyanobacteria which are easily missed in microscopic investigations but are of particular interest for that reason. And we also want to attempt to breed these in the barrels.
The samples captured have to be filtered in the ship's laboratory in order to separate the algae from the water. The filters with the algae on them are frozen and packed for the main investigations using the mass spectrometer at the Humboldt University in Berlin. Gas with heavy nitrogen, 15N, was added to the cyanobacteria. This differs in molecular weight from the light nitrogen usually occurring in nature, which is 14N. In the mass spectrometer, it is possible to measure subsequently whether or not the cyanobacteria have processed the heavy nitrogen.
I am Anja Hansen, and I work at the Institute for Baltic Sea Research in Warnemünde. Here in the Benguela upwelling system, I deal with the phytoplankton and take samples, which we then analyze at the Institute. For this, I mainly take water samples from the CTD Rosette water samplers, which then undergo qualitative analysis, which involves determining the cell number and biomass. I also take samples with a hand net in order to determine the species in the water column.
Bestimmen zu können. Also das Handnetz ist das kleinste Netz, was für die Meeresforschung eingesetzt wird, also zumindest hier auf dieser Reihe.
The hand net is the smallest net that is used for marine research, or that's the case on this trip at least.
Fischen wir damit in einer lichtdurchfluteten Zone.
With this we fish for microplankton in the sunlit zone. We mainly deal with microalgae, which have chloroplasts.
Die Chloroplasten besitzen und die Chloroplasten enthalten das Pigment Chlorophyll.
The chloroplasts contain the pigment chlorophyll,
Zellen in der Lage,
so the cells are capable of converting solar energy into chemical energy to produce biomass.
und dann Biomasse zu produzieren. Denn dazu brauchen sie natürlich auch noch Nährstoffe.
Of course, they also need nutrients for this.
Das sind halt vornehmlich Einzeller, die ich mir da anschaue in den Proben.
I primarily look at single-cell organisms in the samples.
Mit der Handnetzprobe verschaffe ich mir erstmal einen Überblick.
And with a handnet sample, I initially get an overview of what species generally occur in the water column. This is a sort of qualitative sampling. So we're looking at the species composition in the water column.
Die Maschenweite beträgt 55 Mikrometer.
The mesh size is 55 micrometers because the microplankton are in the region of between 20 and 200 micrometers in size.
Mikrometer klein. Und ich schaue mir das im Mikroskop unter 100.
I then look at the cells under the microscope at 100 to 400 times magnification.
Ja, wir sind jetzt hier schon ein bisschen näher an der Küste.
Here we are already a bit closer to the coast, meaning the nutrients increase, the nutrient concentration increases, and as a result we have slightly more phytoplankton in the water column. This can also be seen by the color of the water as well as the catch I have in the net. Now I fill my bottle with a catch.
Die Probe fixiere ich dann.
And later I fix the sample so that we can take it home. Later on, I can take a look at what is in the sample under the microscope. These are the samples obtained with a hand net, which I'm looking at now under the microscope to get an overview of what species are present at this site. Quite characteristically, we have a main biomass of diatoms, which are typical of this time of year.
Die sind für diese Jahreszeit typisch und auch, dass wir jetzt wieder eine höhere Biomasse haben.
It is quite clear that we have a greater biomass here because we are slowly coming back into the coastal area.
Durch das Auftriebswasser werden ja Nährstoffe an der Küste in die Wassersäule.
Nutrients are carried into the surface water on the coast by the upwelling water, and this then becomes a food source for the phytoplankton. This then grows and drifts out into the ocean. It then becomes vitally important for other organisms at the beginning of the food chain. The exact distribution of the phytoplankton, the biomass, from the coast over the shelf into the open ocean is what interests us. It is what we deal with in our research group at the Institute for Baltic Sea Research in Warnemünde.
My name is Andreas Kunzmann.
My name is Andreas Kunzmann, and I'm from the Center for Tropical Marine Ecology in Bremen. I lead the ecophysiology research group there. We are currently at 20 degrees south off the coast of Namibia in the Benguela upwelling system, and we are searching for fish. For this we use our ring trawl, which is a net with an opening of around 1.5 square meters and a mesh size of around 500 microns. We are looking for early life stages of various fish species. Early life stages means larvae and juvenile fish. And like all living organisms, like all animals, fish need oxygen to breathe and for their metabolism. One of the things that characterizes the Benguela upwelling system is that it has zones low in oxygen, or indeed oxygen minimum zones. They come about as the cold, nutrient-rich water comes to the surface and fuels plankton blooms, which, if they are not eaten, die off and are decomposed by bacteria. These bacteria need oxygen for the decomposition process and obtain it from the water column, leading to minimum zones which can sometimes be very extensive. A fish that has to cope with an oxygen minimum zone because it lives or feeds there or because it is carried through it by the current must develop certain adaptation mechanisms, and these are what we are especially interested in. In order to be able to carry out specific experiments with the fish, we have brought a laboratory container on board with us, which has certain equipment that enables us to simulate the low oxygen content of the upwelling system. This means we provide the fish with very specific oxygen concentrations in the water, and we observe how it copes with these oxygen conditions. Firstly, we record the metabolism in the form of oxygen consumption, and secondly, we record activity. For this, we use a video camera which records the fish around the clock for 24 to 36 hours. Here we are currently in the middle of a respiration experiment. We want to know the oxygen consumption of a fish we have here in the measurement chamber. In this case, it is a goby, which is a demersal and is known to be able to tolerate very low oxygen levels. We want to know more about how low the oxygen level can be without the fish getting very stressed. Using a computerized system, we are able to regulate the oxygen content between two limits and at the same time measure the oxygen consumption of the fish. We are keen to provide the fish with as natural conditions as possible, so we have installed a daylight lamp, which is on for 12 hours and then off for 12 hours, meaning we use a day-night rhythm for these measurements. To allow us to film uninterrupted overnight, we also have an infrared lamp, and we close this tent at night so that it is nice and dark and the fish is completely undisturbed. With the aid of the infrared light and the camera, we can just carry on filming the experiment at night and record the activity of the fish. The results of the oxygen consumption measurements so far and the tolerance give us relatively good information about what fish can tolerate, what they have adapted to, and with the help of the figures, we are able to make predictions about where we can find certain types of fish.
In addition to the physiological tolerance limits relating to low oxygen content, the oxygen consumption measurements carried out on fish larvae also tell us something about their nutritional requirements, in combination with stomach contents analyses, which tells us the dietary composition, and chemical analyses of different food organisms, which determine the different energy content. With this information, we can then convert the oxygen consumption into the daily nutritional requirements. Our results show that young life stages of various species are very different both in their physiological tolerance limits and in relation to their feeding ecology, and both are factors that can have a critical impact on the reproductive success of fish species. For example, in my doctoral thesis, I have been able to show that the larvae and young fish of horse mackerel are very resilient to external environmental influences. On the one hand, they are very tolerant of low oxygen content, but they can also successfully exploit food resources under a wide range of environmental conditions. These are two explanations as to why this species is able to reproduce successfully even under the changing environmental conditions in the North Benguela system and still represents a very important fishery resource.
In the Genus project off the coast of Namibia, the research group led by marine biologist Holger Aul is investigating the zooplankton in detail. The aim of the work is to thoroughly survey the crustaceans present in the area. How many different species of copepods and decapods are there in the Benguela upwelling system? And what are they? To catch enough examples, the researchers from the University of Bremen use a multiple opening-closing net.
This allows us to collect samples from various depth layers so that we know exactly which depths the animals come from. This is important because we want to do experiments with the animals, and for this we need the precise water temperature in which they occur in the wild. This is why we must know what depth they're from. On this trip, we can also benefit from the work of the other study teams that deploy different nets, especially with the decapods, the shrimps, and the larger crustaceans. They are so scarce that they aren't normally caught with our small net, and we get them out of the bigger nets from the other study groups.
For example, this large specimen of a decapod was caught at night with a ring trawl. The researchers believe there are around 10 different species of decapod in the Benguela system.
So far, we don't know yet exactly what species it is, but we have identification keys, and now we will try to determine the species. To identify a decapod, you can look at various features. You can see whether these segments overlap or not. Then you can see whether the legs have pincers or not, and if so, how many. Then you can look at the shape and size of the eyes. And you can also look at whether there are any points or teeth present at the end of the body.
Copepods and decapods play an important role in the ecosystem because they occur in large numbers and represent an important food source for larger organisms. A key objective of the Genus project is to clarify what copepods and decapods themselves feed on. To find this out, the researchers must carry out extensive studies, partly in the laboratory in Germany.
You could actually investigate this with classic methods in which you either run feeding experiments or look at what the animals have in their stomachs. There is, however, the problem that this is always only a snapshot and can be somewhat biased by what the animals eat in the end of the net, where they are very close together while being captured. This is why we investigate it with what are known as trophic biomarkers. These are either fatty acids or stable isotopes which are taken in with the food and incorporated into the body tissue. You can basically sum it up with the phrase, "you are what you eat," which means nutritional components are incorporated into the body matter, enabling us to draw conclusions about the animal's food.
The key questions for the researchers in the Genus project don't just concern food composition. In order to be able to make statements about how much the animals eat, studies into food quantities are also carried out.
There are a variety of methods. The obvious method would be to carry out feeding experiments. In other words, the animals are fed. However, this is relatively complicated. You must always offer the right food, but you cannot provide the natural food. This is why we have agreed as part of the Genus project that we will not determine the feeding rate directly, but instead take oxygen consumption measurements, known as respiration measurements, and afterwards calculate food intake from the oxygen consumption using a mathematical algorithm. This is basically the same as for us humans. We have to breathe because we burn our food with the oxygen we absorb in breathing, and it's exactly the same for crustaceans.
Biologist Rolf Koppelman and his team are working in the Benguela upwelling system off the coast of Namibia. The main research focus of the scientists from the University of Hamburg is on zooplankton.
We are investigating meso and macroplankton in our project. Meso and macroplankton are different size groups of zooplankton ranging from a few hundred micrometers up to several millimeters. Plankton comprises all free-swimming organisms that are not able to actively move against currents. This spectrum includes mainly smaller crustaceans and other organisms, but large jellyfish, which also drift in the water and cannot actively move against the current, are included too.
Nowadays, optical and acoustic measuring devices like video plankton recorders or sonar systems are used to detect plankton. However, to obtain data about the taxonomic composition and spatial distribution of the zooplankton taxa, and to gather material for biochemical investigation, additional catches are made with various nets.
If you want to know exactly which organisms are there, what types are present and how they are distributed, we have to do what our ancestors did, fishing through the water with nets and bringing the animals up to the surface. We now have more modern nets. We use a so-called mock net. It's made up of two net systems which are next to each other and have an opening of 1 square meter. The systems have 9 nets on each side which can be opened and closed one after another. We simultaneously record all the environmental parameters like temperature, salt content, depth and flow rate and the amount of water filtered. This allows us to sample the water column in small depth intervals.
The organisms in the Coden buckets are fixed with formaldehyde or frozen. They are then weighed in the laboratory on land and sorted according to size. Then the project's planktologists determine the animal's group and species.
In order to investigate the position of the organisms in the food web, we carry out further analyses. For example, we investigate the composition of stable isotopes of nitrogen and carbon in the animals and carry out gut content analyses of individual animals to examine what they have eaten. This means we can later understand the role of specific organisms in the whole system over long periods. Ketognaths or arrowworms play an important role in the zooplankton community. They are predators, measuring up to 12 centimeters in length, and they feed on other zooplankton like copepods, for example. They can be found in the upper layers of the water column around 100 meters, but they can also be present at depth of several thousand meters.
As an addition to the plankton research, the sediment structures and fauna living on the seabed were recorded at several study sites as part of an expedition with the research vessel Maria S. Merian in March 2011. A remotely operated underwater vehicle, in short ROV, was used by the University of Hamburg. This device can dive to 1,000 meters depth and is equipped with various sensors and cameras. It is connected to the ship via a fiber optic cable and is controlled from there. The sites investigated were very heterogeneous in terms of sediment structure and population diversity. Off the coast of Namibia, the Benguela current brings nutrient-rich water from great depths to the surface. These kind of so-called eastern boundary upwelling systems are highly productive and supply a large part of the world's fisheries yield. Scientists from the Leibniz Center for Tropical Marine Ecology are investigating the qualitative and quantitative composition of the fish population in the northern Benguela region and observing a change in the species distribution.
There
have been significant changes in recent years. Usually, it's the sardines and anchovies that are the primary predators in these upwelling systems, which eat all the plankton. small zooplankton and the phytoplankton, and used this basic food source to build up large stocks. But then in the 80s there was intensive fishing in the area. It was at a time when Namibia still wasn't independent. Foreign nations fished in these waters, especially for sardines and anchovies. So the stocks of these fish were significantly reduced, and the problem today is that the sardines have still not returned. The current stock is very small, yielding 10,000 tons per year. This is close to nothing compared to the 300, 400, 500,000 tons that used to be in the good old days. And the question is why they don't show up again.
The commercial fisheries is now reliant on horse mackerel and hake. In addition to overfishing, environmental changes are also most probably responsible for the decline in sardine and anchovy stocks. The Bremen scientists' investigations concentrate on the early developmental stages of commercially important fish species.
Within the life of a fish, the larval and juvenile stages are the most vulnerable to any environmental changes there may be. Fish larvae are planktonic. This means they cannot choose freely where to go. They are not capable of swimming long distances independently. They drift with the water. This means if they find themselves in poor environmental conditions, they can't really escape. Larvae are also more sensitive than large individuals, simply because they are more fragile and small, and not all of their life functions are perfectly formed yet.
Deterioration in the environmental conditions, for example, a change in water temperature, oxygen distribution, or the concentration of nutrients, can lead to a change in the community of microscopic plankton species. These serve as food for the fish larvae, so this could have a considerable influence on the development of the fish population. The project is investigating the position and importance of ichthyoplankton and juvenile fish within the food web, and the extent to which the different species are influenced in their development potential by changing environmental conditions.
Of course, it could be that there is simply not enough food present, for whatever reason, meaning that the fish larvae begin to starve and the probability of surviving is therefore relatively low. The other possibility, especially interesting and problematic in the Benguela system, is the extension of zones in the water in which oxygen content is very low. In the Benguela system, there is also a gas toxic to all organisms called H2S, hydrogen sulfide, which comes from decomposition processes on the seabed and which produces from time to time bubbles raising through the water column. It extracts oxygen from the water and is toxic by itself. If it encounters fish larvae or fish eggs, of course, it's not good for them. The question we investigate is whether the various species cope differently with these difficult conditions.